Hello, everybody. I want to talk about today things that we should be studying in the medulla and the reptile cerebellum because it's very fascinating. And it's interesting and just the ways that we can study this, because I looked at the ball python compared to the boa. So we have a ball python here. We can see that this is thicker. This medulla is thicker than the actual and has more of a hump than the rainbow boa. And this is the cerebellum. They pretty much look very much alike, the cerebellum. And the cerebellum controls a lot of the regrowth, the muscles, and things like that. But everything has to go through your cerebellum or your medulla. And the medulla has a lot of the hormones and stuff in it, which is really fascinating to me. So this is a bearded dragon. This is the medulla. This is the actual cerebellum. The green is cerebellum, the yellow is the medulla. Okay. So this just kind of shows you where the brain actually goes into. So why does the medulla kink up like that and thicker? Not the cerebellum, but the actual medulla. Why does that do it for the ball python and not that? I looked at anoles and I made a mistake in the Dylan Parrish video, Animals at Home video. I showed a tanny lizard, a tanny dragon instead of an anole brain, and it had the kink. See, this is actually the tanny dragon. This is a massive kink here. And this right here has to be the cerebellum. This is a tammy dragon. It's a little bigger than an anole, maybe something like an emerald swift or something size, which is very interesting. I mean, it's a super cute little thing, but not that big. Now, when I found anole brains. So right here, we can see this is more like a beardy. I think it might be kind of thick. The actual deal may be thick, but here's your cerebellum right here for the anole. And here's your medulla coming out. But it doesn't have the kink. Yeah, there's no kink according to these deals. So that's very interesting to me that they don't, because they should have very tactile senses here. But we have to compare size. So I finally found a gecko something or other. And this does have a very thick medulla and a very thick cerebellum. So this is a gecko, a thick-toed gecko. And this is what I was talking about. The sensory down here in their toes would have to go up and come through the medulla into the cerebral, come back out, go to the optic tentum, and then come back down and go to the forebrain. To think about it, that's a lot of information just moving around everywhere. And it does have to be linear. It's not a human brain where the cerebellum and this part in here is the medulla. It's not spherical like ours is. So it does have to work in a moment, and I'll get into that. We'll go over blind sight and that eventually, and I'll explain what blind sight has to do with the reptile brain, because it does show us that these brain signals are linear. They don't just magically appear. So looking at an anole, the anole has a lot of the same things a gecko has, or something similar in the way they touch things. And that's why I wanted to look at the anole brain. And this is fascinating. This is him simulating a hurricane. A normal one, and in this really showed us that what in this guy's field experience, because of the hurricanes that are happening more often, these anoles in his career lifetime, so within 30 years or less, the anoles have actually grown longer fingers and longer front limbs to be able to hold on to trees and stuff during hurricanes. A truly evolutionary feat. Now, this feat would have to have something to do with the medulla and the cerebellum, which is very fascinating to me. And I would be very curious to see this different anole's brain over a regular green anole. Is there a difference? Because that tactile sensation should go through the medulla, which is really cool. So that's something to study right there. Study the difference in the medulla and the cerebellum of that particular lizard, of anoles compared to other anoles. This is a very important subject matter. This has to do with evolution of muscles and bones. Very interesting. So here's the case here. Marine iguanas. It's very interesting. I love the skull makeup of it. The way it does. So they're measuring him or it. And then they're going to x-ray him. So it's believed that the marine iguana from the Galapagos Islands, stuff that Darwin hated, actually shrink during certain times of the year where they can't get to the food source of the actual marine plants. So therefore, they allow themselves to shrink to take up less energy and then grow. Well, this right here is going to actually be affected by the medulla and the cerebellum. Now, the cerebellum and medulla here, this could potentially have massive impacts in the medical community of humans with thyroid problems, because those are where your glands are often at, is the medulla. The medulla should be studied on this animal, and I don't see that. So this marine iguana is really cool, but here's an abstract on the marine iguana, or a study that was done on their bones. So it really goes over a lot of the bone structure. And we can see in this picture right here, it's showing us the frontal. This should be where the actual forebrain is. The front brain is right about here. Now, we can find this picture of an actual iguana's brain. So this part right here should be fitted in right here, this blue. And so these parts right here are the optic nerve. This is the actual nasal, the oral stalk that I talk about. So we can see here, you can see the separation right here. There's some white matter here, some gray matter between these two, which is very interesting. I love this. So this is suggestion that maybe iguanas can have more potential for emotional intelligence because of this white matter between this gray matter. Because that's what this is the gray matter. This is the optic tantum. And the connection between these two is my theory of why humans have empathy the way we do. Because the modeling part of empathy is in the optic tantum, and our empathy is probably a portion of our imagination. So if this could make that connection, I'm not for sure because it's not spherical, but there's potential. But right back here is the cerebellum, but there's really no kink. But this is an iguana, so they're probably talking about a green iguana here, not a marine iguana, which does make a difference. So it would be very interesting to see the bones, the iguana itself's neural pathways, and let's see if we can get a back image of the back of the skull right here. So this is where the spinal cord and the medulla will be here, right here in the middle. And so there could be a kink. There might not be a kink. We don't know. But there's enough room for there to be a kink when the brain's in here at the top, right behind the eyes. So that's a very fascinating thing to me. We can see right here, this is just a iguana, like I said, right behind the eyes. And it goes down. There's no reason to believe this is any different for the marine iguana. If we found the marine iguana was different, that would make a massive difference here to actually study this. And when we study the medulla, we want to look at cells. So we see the adrenal glands is something to study in the medullas here. And this is what I'm talking about. So there's different types of cells. There's two types of cells that I know of that they've talked about. And these differences in cells is something that we should be looking for. I don't want to go too much into that. I don't think it's something that most people would really want to learn, but it is something that we absolutely have to look at, is the cellular structure of the medullas. And like I said, this has massive potential in people. Now, as I'm recording this, there's a guy from Australia that I talked to about Peter-banded skinks, and the Tanny dragon actually has a very interesting body languages and stuff that it uses. It uses its tails and different things like that they've studied. I'll try to include something in that, but maybe that's why the medulla is a little different. But that's very interesting that that's the case. It's so much of this we don't know. But as we can see, thyroid glands, all these different glands that we have in our medulla that could be affected. And then the cerebellum has control over our muscles and bones and stuff like that, and regenerative processes. And both of these connect. That would create the marine iguana to change size. That's where one of those processes would happen. And so that's why that has to be studied in that. And then we might be able to apply that to actual human medication to where we could actually lower thyroidism or any other glands and have a more massive impact on that. Now, back to the ball pythons. So what we should be doing is studying the cells that are different in ball pythons and rainbow boas. You should be able to find a dead python, ball python, and dead rainbow boas all the time that a biologist And this could be an undergrad study in a way, maybe more of a master's study, that you could actually chop that brain open and see this stuff. It would be very, very interesting to see that difference. Here's another study that I thought of that we should do with ball pythons and rainbow boas and such. Let's see, because the tactile function of a gecko and anole should have some sort of effect on the medulla. Now, the one thing that we know of is when things crink, it's because they're gaining more surface area. And that's why I believe, so if the anole's brain doesn't have that kink, but yet the medulla is bigger than most other species of the same size, then we could make the same hypothesis that there's more cells there to take more of that information. Whereas the ball python may need more cells because they bury themselves in termite mounds. So they would actually have an increase in tacticity, being able to touch, than other snakes might that just climb trees. That's a potential there. And so where it needs to be bigger, it may not be able to have the capacity to get bigger, so it creates a kink to create more surface area. And so that way it has that deal. And that's what we should be studying in the cells. So if this is right and they have more tactile functioning, then one way we could actually test that is if we take an enclosure, we let them escape, take that same enclosure, turn it around, and see if because they have memory. This has been proven beyond a shadow of a doubt. To give you an example, so even though I'm moving my fingers on this side, she should be going after them, but Pitch is smart enough and remembers that the other side of the enclosure is where she gets fed and she is hungry. Look, she doesn't go after my fingers. And later on that day she did, but so she can't understand glass no matter how dirty it is. This was taken on the quick, but here in just a second, she's about to slap the glass because she knows this is also where I get out. See, she hits her nose on the glass because she thinks she can get out. If the animal's trying to escape, it should actually be able to memorize the escape. So is the memory from what it sees, or is the memory from what it touches? Because the acrylic's going to feel different than the rest of the actual enclosures. So then, like I said, you take an enclosure, you let it escape, you turn it around, see if it changes the way it escapes, if it tries to go back to the original, if it feels around, how does it do it, and does it find the escape? And then you need to build the same size enclosure completely out of acrylic and see if it'll escape then. And another thing you might be able to do is etch a little X or something, a circle, something that the snake can touch in the acrylic that there's no way it can actually see, that it can understand. This would be very helpful in understanding if their tactile function of a ball python is different. And then we have a way of saying, okay, so this may be an actual theory on why the medulla is different. See, these are studies that can be done often under PhD. Doesn't have to be a PhD. Some of these studies we can do at home. And some of these studies will have massive implications on medications. This is why the cerebellum and the medulla are so important to study. This is why we do this. This is what we need to be pushing, because it has the potential to affect us. Thank you all for listening. I would love to hear what your thoughts on all this are. What do you think we should do next? What are some of the studies that you think we could do on the medulla? Do you think that I might have gotten something wrong? I'll include all the links below. Love to hear back from you guys. You guys have a great night.